Eric Topol here for the Genomic Medicine Resource Center, and the topic today is smoking, lung cancer, and peripheral arterial disease. This is a fascinating set of articles that were published in Nature and Nature Genetics in early April 2008. Three papers, one from the Icelandic group, a second from the group of Hung and colleagues from France and throughout Europe, and the third paper by Amos and colleagues from the University of Texas MD Anderson, as well as multiple colleagues in the UK and internationally. These three studies with remarkable concordant results regarding cholinergic receptor subunit genes, multiple subunits, were found to be linked unequivocally with lung cancer. There were some disparate results by nature of the details of phenotypic or demographic data, particularly with smoking. As one would expect, these genes that are cholinergic genes that have been tied, acetylcholine, with smoking addiction in the past, one would think that the link is actually through smoking and not directly to lung cancer. That's actually where the three studies differed in some respect. Firstly, all three of these studies were done in very large populations with the Illumina platform. Over 300,000 SNPs were assessed in thousands of cases and controls. All three demonstrated a tight association of the cholinergic subunit genes, CHRN3, 5, and others, with lung cancer. The differences actually related to the Icelandic study, where there was the number of cigarettes smoked, pack years and cigarettes per day. There was a correlation suggesting that the link to lung cancer with an odds ratio of about 1.4 was actually mediated through smoking cigarette addiction. The other two studies were not able to demonstrate that linkage through smoking cigarette addiction. One would think, however, that's likely to be the mechanism, but this requires further dissection. It's actually a fascinating interplay between genes and environment to understand a gene that makes one more susceptible to nicotine addiction and thereby influencing the risk of lung cancer. There was another wrinkle that's very interesting in these three studies, and that was the establishment of the same acetylcholine receptor and likely nicotine addiction pathway to the development and risk susceptibility to peripheral arterial disease, while the odds ratio for that was not as high as lung cancer. In fact, it was 1.19 as compared to 1.40, and only demonstrated in the Icelandic study. It certainly was impressive and significant through multiple cohorts of replication. So now it appears that from these three major papers and contribution, that indeed we have a new link, one that heretofore had been looked at in association study with candidate SNPs, the relationship between the acetylcholine cholinergic receptor and cigarettes smoking ticotine addiction. But that extends now to the direct association with lung cancer, and at least by the Icelandic study, to the risk of developing peripheral arterial disease. So this is fascinating. It's really one of the very first genome-wide association studies that's probed into an environmental issue, and it'd be great to get your thoughts on this series of articles with a company editorial about smoking out the smoking genes in Nature by David Hunter and his colleague. I'll be very interested in your thoughts and hope to see your posts on the Genomic Medicine Resource Center. Thanks very much.